All right, so far we've talked about a whole bunch of different aspects of cancer development, things that need to take place. Let's try and put it all together into a big picture of what sort of the series of events might look like during the development of a neoplasm or a cancer in general. The idea here is that you have to have a series of events take place. So first off, we've got normal cells. And then we've talked about carcinogens. So some sort of carcinogen needs to come in and cause some sort of damage. That all by itself is pretty common. That happens all the time, and people don't develop cancer. So why, then, in some individuals, does this lead to the formation of some malignant neoplasms? Well, this initial state, this carcinogen, is just one of the steps that then needs to take place. So you've got some damage, but that damage doesn't do enough damage to lead to full-on autonomy by these cells. There still needs to be cell cycle dependent either growth factors or promoters or something that's going to cause the cell to undergo growth and differentiation. So we've got some sort of damage here. If that cell just kind of sits around and never divides again, it doesn't really matter. So we need division for that cell to then pass on those damaged genes. And usually that's through the promotion of a division. So we've got like growth factors. Now, maybe if this is part of the reproductive system, the growth factor responsible for that growth is just continued estrogen or testosterone. Okay, so testosterone, those hormones they're present. Maybe it's chronic inflammation. So you've got consistent damage to tissues, and then the growth promoting or healing cytokines that are going to be released by the immune cells. So whatever it is, you have continued growth, that promotion of growth. And each one of these that's being activated, let me switch to a different color, that are being activated here, all inherit that initial mutation, that initial damage. So now we've got this clump of cells that's been growing, and it's only been growing due to factors that keep it growing, the promoting factors. Eventually there's enough mutation that builds up as these are dividing that now, once we get to the progression stage, this now can grow without any problems. So it doesn't need the promoters anymore. It doesn't need any external factors. It's developed enough mutations now that it can overcome the cell cycle and then just continue to grow. This also gets to the idea of why some neoplasms are malignant, will actually cause damage, and why some neoplasms are benign. If we erase that. If we end up staying in this stage where the damage has been induced, the promoters keep it growing, but if those promoters go away, that tissue is going to stop. Like it's got damage, but it's not going to continue growing all by itself. So that's really the difference between benign and malignant, is getting past this last step, the progression, to the point where it can grow independently. All right, we talked about this before, but this is kind of a quick review here. Autonomy versus anaplasia. Autonomy means the ability to grow without regulation. So the ability to just start dividing and dividing and dividing. Normal cells should not be able to do this. Okay. Anaplasia is the differentiation from a stem cell into a specific tissue. In this case, this stem cell into this specific tissue. Now, there is a continuum, and that's what's shown down here. A continuum between the proliferative capacity. So stem cells are highly proliferative, but as they become more and more differentiated into whatever tissue it's turning into, it loses the ability to proliferate, but gains the ability to be very specific in that cell function. So if this was just a stem cell here and it's turning into a hormone-producing cell, as it makes its way down this, it stops being able to proliferate whenever it wants, but it does a better job at becoming a hormone-producing cell. So now it can produce hormones where this stem cell can't produce hormones, but it can't really differentiate where the stem cell can. What cancers do is that they maintain a little bit of both. As they're proliferating and becoming more differentiated, they'll stop maybe somewhere in the middle where they still have the ability to proliferate and they are gaining some of the differentiation. They no longer look so much like the initial stem cell. They're gaining functions, but they don't make their way all the way to the differentiated state. And so maybe they have a whole bunch of different functions here, and they do it all very badly. But maybe they have a whole bunch of functions. Maybe they can produce tons of different hormones instead of just the one type of hormone that they were going to turn into. So again, autonomy, the ability to grow without regulation, so nothing needs to tell it to divide, it's just going to divide. And anaplasia, instead of becoming whatever target cell is, it doesn't fully become that, and it doesn't fully gain the function of that cell. It's not going to be a good hormone-secreting cell, it's going to be a bad hormone-secreting cell that's going to secrete all different types of hormones, not just the one that it should have in this case. I've already mentioned this too, but just again as a review, we can classify tumors as benign or malignant. So benign tumors, generally that means that they're not really dangerous, and then malignant tumors are mal, meaning bad. So these are things that would normally be described as cancer. Main differences here: benign tumors have proliferation, so they have the ability to grow, but they don't lose the ability to differentiate. They stay pretty similar to the surrounding tissues. So they're proliferative, but they're not super anaplagic, or they still maintain their morphology, their shape, similar to the surrounding tissues. Also, usually benign tumors have a good definition between them and the surrounding tissue. So the immune system has done a decent job of walling them off. There's usually some fibrous connective tissue there. In general, benign tumors aren't super dangerous. Now, that can be misleading, though, because there are cases where benign tumors can be life-threatening if, as it's growing and pushing against the other tissues, it causes damage through that. So in the brain, for example, a benign tumor can still kill you if it's putting pressure on the cerebral blood flow, shutting off blood to certain areas, and then leading to stroke and death. Okay, so the tumor not dividing and breaking out and metastasizing, I mean, that's good. Like, benign tumors don't do that, but they can still be dangerous, still cause damage. Malignant tumors, though, are worse. So in a malignant tumor, we don't see that clear differentiation between normal cells and healthy cells. We also see a loss of that differentiation. They don't really resemble the initial tissue very much. So not only are they proliferative, but they're also anaplagic. They don't really resemble things, and they can easily spread or metastasize to other locations because they're not walled off. So malignant tumors don't stay in their location. They'll start to spread, and that spreading is going to cause damage to other tissues. Cancers, again, this is malignant. Benign tumors don't spread. They stay where they are. Initially, in the production of a malignant tumor, you have the local growth, just like we saw above. That local growth, as they continue to grow and grow and grow, once they reach past the initial tissue, let's say if it's an epithelial tissue, once it gets past the basement membrane. If it's a muscle tissue, once it leaves the muscle fibers and makes its way into the connective tissue. That's a move from local spread to a direct extension. So local spread just in the initial tissue. Direct extension moves into neighboring tissues. These neighboring tissues are tissues that aren't the same as the tissue of origin. As that direct extension starts, we can have little extension or little colonies that start to grow in that secondary site. That's called seeding. So this is colony growth in new tissue. That's kind of what we're seeing up here. We have the primary tumor up here. That's the local growth. Direct extension moves into the surrounding tissues right here. And then we get metastacism. In metastacism, we move past the local or secondary tissues and move to a distant tissue. Usually for metastasis to take place, we move through blood vessels, usually the veins, and the lymphatics. These are lymphatics. These are systems that are already set up to deliver materials throughout the body. They're transport systems, so hijacking these is a really easy way to get from one location to another. And in order for this to happen, metastasis to happen, it has to go through a couple of steps. It has to make its way across the barrier here of the membrane, of the vessel, moving through the endothelial cells, make its way into the fluid, transport to a distant location, and then it still needs to exit. So if it stays in the lymphatics, it's probably going to get eaten up by our lymphocytes. It also has to make its way out and then take up residence in the new tissue. So that's a metastasis. The new tissue that it goes to depends a little bit on the type of cancer. So cancer, like your book points out, cancers in the colon will make their way to the liver and take up residence there because there's a direct connection from the portal blood vessels that will deliver them directly to the liver. And then the tissues of the liver are similar enough that they can bind to those tissues and start growing. Other places, you can get locations in the lungs too. So moving from the liver to the lungs, direct blood connection. Moving to the lungs, get stuck there, we can do a metastasis there too. So not all tissues will be favorable for the new metastasis, and so sometimes we can see tissues or areas that are more preferential to certain types of cancers. That's called tropism. So having a favorable tissue to the cancer. And some cancers don't need this tropism. Some cancers can just invade any location and just start growing in that spot too. So that does exist as well. But things like prostate and breast cancers tend to make their way to the bone marrow for some reason. Just a favorable environment that way. All right, let's talk about how we name different types of neoplasms. So any neoplasm can have the suffix oma on it. This isn't 100% that everyone will have it, but we can use that to differentiate. Some of them have special little suffixes too, but in general, like oma refers to a neoplasm. Next thing we're going to look at is where this tissue started. So the tissue of origin, we'll usually go towards the front of the name. So if it originated in the epithelium, this would be an epithelioma if it was benign. So tissue of origin and oma, if it's benign, you can just stick those together. So if it's a malignant or a cancer, we also would add carci. So carcinoma together would indicate a malignant or a cancer itself. An epithelicarcinoma, we wouldn't usually say that. We'd get a little bit more specific. But looking at the different types of epithelia, so if it was at the surface, that would be a papilloma for benign. If it was glandular, it would be adeno. That prefix means glandular tissue. So adenoma is a benign tumor of the glandular tissue epithelium. If it's a carcinoma or an adenocarcinoma, that's a cancerous epithelial-derived glandular tissue, malignant, damaging neoplasm. And we can take a look at these other ones too. Endothelium, we've got the blood vessels. Endothelium are those internal cells that line blood vessels and lymph vessels. So if it's from the blood vessel and it's a benign tumor, it would be a hemangioma. If it was a lymphatic tissue, it would be a lymphangioma. If we get into carcinomas with these, hemangiosarcoma, lymphangiosarcoma. Hopefully you're seeing how these kind of break down. The weird thing is when you get into the different types of like connective tissues, it's a fibrous fibroma. If it's lipid-based, it's lipoma. Cartilage-based, chondroma. Bone-based, osteoma. So you just have to know the prefixes for those ones. And then fibrosarcoma, and so on and so forth. So using the oma as a suffix, tissue type to determine where, and if it's malignant, add the carci in there. Again, this is not 100%, but a lot of these will follow this. And something, too, that's not shown up here is just the term in situ. In situ is usually used for malignant tissues that haven't left the primary area first. So they haven't metastasized yet, they haven't moved into even the secondary, but an in situ glioblastoma is something that's still in the neural glia and hasn't made its way out. That's a good sign usually. Means you probably caught it pretty early.